Hello friends, welcome to discussion on this topic Introduction to Dosage Form, Classification of Dosage Forms and their definitions. The key learning objectives of this particular topic shall include the process of drug development, a typical drug discovery cycle, classification of dosage forms, definitions of dosage forms along with examples. First of all, let us see what is the process of drug development. Drug development is a process of bringing a new pharmaceutical drug to the market once a lead drug has been identified through drug discovery. It includes preclinical research on microorganisms and animals, filing for regulatory status and permissions to regulatory authorities for investigational new drug to initiate trials on human. The drug development process may also include additional documenting and process of obtaining regulatory approval with a new drug application to market the drug. This is a typical drug discovery cycle. Initiates from either identifying a drug from natural origin, that is animals, plants, minerals, and collecting of the compound. Thereafter, these compounds are subjected to primary assays. These are high throughput assays, in vitro assays, in order to determine the identification and purity for that particular compound. Alternatively, a lead or a new chemical entity is identified for a particular target or a biomarker. These lead chemical compounds are identified either through design. There are specific softwares like computer-aided drug design that helps in screening a large number of chemical compounds to select the right suitable candidate for a particular target. Not only that, they also help to identify the right structural characterization of a protein ligand complex that can work as a specific lead. Once the lead chemical compound is designed through a software, they are synthesized chemically by a synthetic chemist, manufacturing chemist in the lab. Thereafter, either from a natural source or from the lab source, these isolated chemical entities are subject to primary assays. Upon successful completion of primary assays results, they are subjected to secondary assays that helps not only to determine toxicity and metabolism, but also it helps to determine bioavailability. Thereafter, during secondary assays only, the physicochemical characterization of the lead is done. Their study in animal models or in vitro cell lines is done in order to establish their toxicity metabolism in animal models or bioavailability in cell lines. The successful candidate is then applied for an IND, that is investigational new drug application is applied to a drug regulatory body in order to move to the next stage of clinical trials. Upon successful completion of phase 1, the molecule moves to phase 2. Thereafter, after successful completion of phase 2, all the documents along with the reports are submitted to the regulatory authorities to obtain permission for phase 3. Upon completion of successful phase 3, the candidate The documentation, along with the report for that particular drug lead candidate, is again filed with the drug regulatory authorities in order to obtain a license for marketing the product. This file is also known as New Drug Application. The preclinical stage covers various aspects to determine physiochemical properties of new chemical entities, their chemical structure, pKa, pH, solubility, stability, polymorphism. Preclinical phase also includes process optimization to transform from a bench scale of a few milligrams in lab to kilo or ton scale in the commercial stage. It should further be examined for its suitability to be made into a suitable dosage form, whether to be manufactured into or formulated into a capsule, tablet, aerosol, suspension, or an injectable dosage form. Together, these processes in preclinical development is known as chemistry, manufacturing, and control. Thereafter, major organ toxicity is studied in animal models. Organ toxicity is studied in heart, lungs, kidney, liver, brain, and digestive system. Not only that, it is also studied for toxicity effects on different parts of the body, such as skin. There are several in vitro methods for carrying out preclinical studies in isolated cells. Many of these tests can only be done by using experimental animals, since it is only in these intact organisms that a complex interplay of metabolism and drug exposure on toxicity can be examined. The preclinical study report, together with chemistry, manufacturing, and control, is submitted to the regulatory authority to move to the next stage of clinical study in humans. The entire preclinical phase documentation is submitted to the regulatory authorities for, and it is also referred to as investigational new drug application or filing to initiate trials in humans. The clinical phase includes phase 1, phase 2, phase 3, and phase 4. Upon successful completion of preclinical phase and upon filing all the documents and the reports with the drug regulatory authority and obtaining a license to initiate clinical phase study, the following different phases are involved for a clinical phase study, that is phase 1 trials. Phase 1 trials is usually performed in healthy human volunteers to determine the safety and dosing. Upon successful completion of phase 1 trials and a predicting response for the particular lead, the documents are filed with the regulatory authorities to obtain permission to move into the next phase of the clinical study, that is phase 2 trials. Phase 2 trials are done to get initial reading of efficacy and further explore safety in small number of sick patients. Thereafter, a successful phase 2 documents and reports are filed to obtain permission to move to the next stage of clinical study on wider population of patients, that is also known as phase 3 trials. These are large pivotal trials to determine safety and efficacy in sufficiently large number of patients. Upon completion of successful phase 3 trials, the documents and the reports, along with chemistry, manufacturing, and controls, are filed with regulatory authorities to obtain permission to market and they obtain license to market that particular drug. The filing procedure is known as new drug application for registration and marketing of that particular chemical entity or a biological molecule. Upon obtaining permission to market the drug or obtaining registration, the products are marketed and distributed. During the post approval or post marketing phase, phase 4 trials is done. Phase 4 trials are post approval trials known as post marketing surveillance studies. They are done by the doctors and physicians to monitor the adverse effects and side effects which are more significant and pronounced in wider populations. So phase 4 trials is reporting of all the side effects which were not observed up till phase 3 or were subdued till phase 3 because the fact it was carried out in smaller patient population. Phase 4 gives a more extensive overview of the clinical effects, side effects, and adverse effects on specific patient population, different age groups, different races. All the healthcare professionals, including the pharmacists, the doctors, and nurses, should actively monitor and be a part of this phase 4 trials for any new drug that has been introduced into the market. There are different sources of drugs. In olden days, there were primitive medicines that were identified from the bark of a tree. For example, quinine was discovered from Africa used for malaria. Lime juice was discovered as a source of vitamin C for scurvy. Plants were identified as a source of drug. For example, ricin from Rauwolfia vomitora, digitalis from foxglove, and opium from poppy plant. Not only that, animal sources were also identified, which includes insulin from pancreas of pigs and cattle. There were mineral sources of drugs like potassium chloride, magnesium chloride. There was chance discovery or serendipity. For example, penicillin was discovered as a chance discovery by Alexander Fleming. Currently, most of the drugs are synthesized in laboratories with a very few from natural extractions. So first of all, let us see what is a pharmaceutical dosage form. A pharmaceutical dosage form is defined as the physical form of a dose of a chemical compound that is used as a drug or medication intended for administration or consumption. So these are the different physical form of the drug or the new chemical entity or the active pharmaceutical ingredient with therapeutic application. They are intended for administration or consumption through these physical form. This is the solid physical form, that is tablets, capsules. This is the liquid physical form of the active drug, that is liquid, eye drops. This is also a solid physical form, that is powder. This is a semi-solid physical form, that is an ointment, paste. This is again a liquid dosage form, that is a cough mixture, syrup, elixir. This is again sterile dosage form, liquid dosage form. Sterile dosage form is injectables, large volume and small volume parenterals. This is aerosol or gaseous dosage form. What is the rationale for drug development? What's the rationale for dosage form development? Why is it important that that particular drug or active pharmaceutical ingredient be formulated as a dosage form? These are the following reasons. Development into a dosage form aids as a mechanism for safe and convenient delivery of accurate dose. For example, tablet, capsules, and suspensions aid in a safe and convenient delivery mode for a particular drug substance. Not only that, a dosage form protects a drug from environment, that is the destructive effect of oxygen or humidity. Example is film coated tablets. The tablets are coated with film coat that prevents the moisture ingress into the tablets. Formulation of a drug substance that gets degraded in the gastric acid of the stomach. They are formulated as entry-coated tablets that helps to prevent the active drug substance from being degraded in the stomach. Entry-coated tablets, entry coating on the tablets prevents the tablet from being destroyed in the gastric acid of the stomach. Formulating a drug substance as a drug product helps to conceal bitter, noxious, and salty taste of drug. For example, sugar-coated tablets like ibuprofen tablets are sugar-coated in order to mask their bitter taste. They are also formulated as capsules. For example, antibiotic powders are formulated as capsules, hard gelatin capsules, in order to conceal their bitter, noxious taste. Formulating a drug substance to drug product also aid in making a dosage form of insoluble drug particles. The insoluble drug particles are suspended as suspensions. Suspension dosage forms are a dosage form of choice for pediatrics. They aid in formulation of volatile oils, fats, fat-soluble vitamins as emulsions. Not only that, there are certain specific drug substances like riboflavin. They are more bioavailable when formulated as emulsion dosage form rather than being formulated as a tablet dosage form. Formulating a drug substance as a dosage form, a controlled release dosage form with large amount of polymer helps to control the rate of release of the drug. Example, sustained release tablets. The other rationale is they help to provide optimal drug action from topical administration. For example, ointment, paste, creams, patches, etc. Dosage forms can be classified in two ways. One is through the various routes of administration, and second is based on their physical form. Here we shall study the classification based on their physical form. First is solid dosage form. A solid dosage form can be of two types, internal and external. Internal is meant for ingestion or consumption. External is for external use. Second type of dosage form based on the physical form is semi-solid dosage form. They are also of two types, internal and external. Third type of dosage form is liquid dosage form. They are also internal and external. The other types of dosage form include sterile products, gaseous. Gaseous products are aerosols and sprays, and all the products which cannot be classified based on the physical form in either of the above categories which we have discussed are collated and together classified into miscellaneous dosage forms. They are novel drug delivery systems, which includes all those dosage delivery systems which are there in research and discovery, liposomes, microspheres, nanoparticles, monoclonal antibodies, microcapsules, osmotic pumps, ion exchange resins, implants, transdermals. The solid dosage forms, as we have studied, are of two types, internal and external. Internals include powders, tablets, capsules, triturates, pills, and lozenges. They are all meant for consumption and ingestion. The solid dosage form for external use is dusting powder. Let us study a brief definition of all these solid dosage forms. Powder dosage forms are medicament with or without excipient in fine state of subdivision. It is supplied either as bulk or individually wrapped in a paper. The individual unit dosage form of a powder can also be supplied in sachets. For example, all the probiotic lactobacillus powder which is available as a single sachet preparation in the market. Not only that, powder for oral suspension are bulk powders that are dispensed in bottles. They're all antibiotic powder for oral suspensions meant for administration to pediatric patients. Tablets are solid dosage forms in which the medicament is compressed in different shapes and sizes. They may either be uncoated or coated. In case the active ingredient is susceptible to moisture and is hygroscopic, then a film coating is necessary. In case they are susceptible to degradation in the stomach by gastric juice, then an enteric coating is necessary. Capsules are cylindrical shaped gaseous made of gelatin which is hard and flexible and are made in different sizes. The blend powder or small particulates or multi-unit particulate systems or mini tablets can be encapsulated in these hard gelatin shells. Liquid dosage forms are also internal and external. Internal liquid dosage form examples are solutions. All the active drug substances which are soluble in the vehicle can be prepared as solutions. Suspensions, in case the active drug substance is not soluble in the vehicle, the particles remain suspended in the vehicle or the medium continuous phase. Emulsions, in case both dispersed phase and dispersion medium is liquid. The emulsions are prepared in two ways, oil in water, water in oil. Elixirs, they are hydroalcoholic sweetened preparations. In case the active drug substance is not soluble in the vehicle or the continuous medium, elixirs are prepared wherein the active drug substance is dissolved in alcohol. Syrups, they are also liquid dosage form which are meant for ingestion. They contain high proportion of sugar in water. Spirits contain high proportion of alcohol. They are meant to be prepared as surgical spirits or, or disinfectants. Mixtures, different types of cough mixtures are example for this. Drafts, drafts are nothing but a single unit preparations of the liquid dosage form. Externals are irrigations, lotions, mouthwashes, nasal drops, ear drops, enemas, and paints. Semi-solid dosage form includes gels, jellies, pessaries. Gels are nothing but a dosage form in which insoluble substances are presented as suspension in colloidal state in a hydrated form and are known as gels. Jellies are non-greasy semi-solid preparations containing a high proportion of gelatin, gum, or starch. Pessaries are medicament that is either compressed in a suitable shape or molded with the help of a base so that it is released when inserted in the vagina. This is one example of a marketed preparation of a particular gel. This is a surgical gel. Other examples of semi-solid dosage form includes ointments. These are semi-solid meant for external use. The medicament is incorporated in the base, usually comprising of greasy substance. This is an example of marketed preparation, that is triple antibiotic cream. Creams are highly viscous oil in water or water in oil emulsions meant for external application. Medicament may also be incorporated in them. Paints are solutions in viscous vehicles or suspensions that are meant to be applied to the skin or mucus with a soft device, such as a brush or a cotton. The other examples of semi-solid dosage form include pastes. Paste contains a very high proportion of solid medicament in relatively small proportion of the base meant for external application. This is one example of a medicinal marketed paste. Confident. Next is suppository. They are semi-solid to solid molded preparations meant for insertion into the rectum. This is one example of a marketed preparation, Dulcolax. This is a laxative. Next is poultice. They are thick and pasty preparations. They have a good heat retention property, so they may be applied on a dressing while hot and bandaged on the affected part to relieve inflammation. Let us now study the various examples of liquid dosage forms. Syrups. They are concentrated solutions of sucrose or any other sugars. Mixtures. They are solutions and suspensions meant for oral use. The vehicle is usually water. Lotions. These are preparations in solutions or suspensions form to be applied to the affected part without friction. The other examples of liquid dosage form includes liniments. Liniments are emulsions or viscous, oily, or free-flowing solutions of drug in alcohol. They are meant for application to the skin, usually by rubbing. Irrigation. These preparations are used as antiseptics for washing the urinary bladder or vagina, the solution being introduced through a soft tube. Drafts. These are liquid oral preparations packed as a single dose in separate containers and in large volumes. Yakult is one example of draft preparation. The other examples of liquid dosage forms are elixirs. They are sweetened, clear, and colored aromatic hydroalcoholic liquids. Example is paracetamol pediatric elixir. Enemas, these are drugs in solution or in dispersed form meant for rectal administration. Ear drops and nasal drops, the solutions of drugs meant for dropping in the ears or instilling in the nasal cavity with the help of a dropper. Now, let us discuss the various types of gaseous dosage forms. First is aerosols. They are suspensions of fine, solid, or liquid particles in gas. This preparation is a pressurized preparation. These are dosage forms for spraying in solution, suspension, or emulsion form with an atomizer device or nebulizers. Typical examples includes all the aerosols inhalers, all the corticosteroid inhalers, Volini sprays. Not only that, other preparations like deodorants, inhalations. These are preparations meant for relieving congestion in the throat. These preparations are either volatile and inhaled directly or contain volatile substances that can be inhaled by adding preparation to hot water. Now, various types of camphor oil, eucalyptus oil are prepared as soft gelatin capsules that are meant to be opened, added to water, and then the steam of that water is inhaled. Sprays. The drug dissolved in alcoholic or glycerine media and sprayed in the form of fine droplets to the mucus with the help of spraying devices. Novel drug delivery systems. These are nothing but miscellaneous dosage forms. So first of all, let us see what is the rationale for development of drugs into these dosage forms. What is the purpose and rationale behind novel drug delivery system? Dosage forms are prepared as NDDS to improve therapeutic effectiveness of the drug substance formulated as these novel drug delivery systems aids to reduce side effects and toxicity. They help or aid to target at a specific site. They also aid in exploring new pharmacological action. These are the various examples of new drug delivery systems. Liposomes. They are artificial microscopic bilayered vesicles or sacs made of phospholipids and enclosing an aqueous compartment. The aqueous compartment contains the drug substance or a drug substance with polymer. This is a bilayer lamellar vesicle made of phospholipids. Drugs can be incorporated in liposomes and administered into the body and can be delivered at the desired site and in desired concentration without being toxic. Examples are doxorubicin, this is anti-cancer product, amphotericin B, gentamicin. Next is microspheres. These are small solid particulate carriers containing dispersed drug particles either in solution or crystalline form. Microcapsules are made from natural and synthetic polymers and administered by injection or through nasal route. Examples are again anti-cancer products like 5-fluorouracil, cisplatin, enzymes, and proteins. Next NDDS is nanoparticles. They are colloidal particulate systems in the submicron size. They are acting as carrier to drug molecules. Their size varies from 10 to 1000 nanometer. These carriers are solid spheres and the surface is amorphous and lipophilic with a negative charge. As a drug delivery system, they enhance the therapeutic efficacy of the drug and minimize the adverse effects on and toxic reactions. Major applications include in cancer treatment and vaccinations. Osmotic pumps is another type of new drug delivery system. They exploit the tendency of a fluid to equalize the concentration of substances on both the sides of a semi-permeable membrane. The system can be designed to deliver different drugs at different rates. The marketed products include phenylpropanolamine and vitamin C. ALSA has recently been introduced in the market, working as an osmotic pump. It has excellent market demand. Next is ion exchange resins. This system involves preparation of drug-charged resin and its drying to form beads. In the GI tract, drug molecule is exchanged for an appropriately charged ion, and hence the drug is released at a controlled rate. Formulating a drug substance as ion exchange resin not only helps to control the release of the drug, more importantly, they help to mask the bitter taste of the drug. The other type of new drug delivery system is transdermal drug delivery systems. These are laminated patches which adhere to skin and permit absorption of drugs from the skin through its layers into the blood circulation. Now, this is in contrast to other semi-solid dosage forms like ointments, paste, creams, and gels, for which it is ensured that the drug or the drug substance from the drug product should not enter into the bloodstream or the systemic circulation. Here, in transdermal, this dosage form is designed to ensure permeation through the skin layers to the blood or systemic circulation at controlled rates, resulting in sustained blood effect. And that is why it is known as a new drug delivery system for dose administration. These are additional informative videos explaining all these new drug delivery systems. We shall see them all one by one. These are the references. Thank you for your time.